episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome everybody again to another episode of our show with another really fascinating guest joining us today, helping to create a better tomorrow for all of us on many different fronts. Today we have the honor of being joined by Dr. Ola Engkvist, who is Associate Director, Computational Chemistry in the Discovery Sciences Research and Development Group at AstraZeneca Pharmaceuticals. Dr. Engkvist is also Adjunct Professor in Artificial Intelligence and Machine Learning based drug design at Chalmers University of Technology. Dr. Engkvist has his Ph.D. in Computational Chemistry from the University of Lund, did his postdoc research at University of Cambridge and the Czech Academy of Sciences. And he joined AstraZeneca back in 2004 and currently leads their Discovery Sciences computational chemistry team within the biopharmaceuticals R&D group, providing computational solutions for all facets of drug discovery. Dr. Engkvist is passionate about pushing the boundaries of using artificial intelligence and machine learning tools in drug discovery. A key focus for him is on building both the team within biopharma R&D and collaborating with various external experts to advance innovation on this front, both in drug design and synthesis. He has a rather pioneering collaboration with University of Munster, where their team demonstrated the first application of recurrent neural networks to molecular design, which has been published in two major highly cited articles. And these methodologies ultimately allow their team to design various novel drug molecules using machine learning to ultimately navigate the very broad spectrum of chemical space that's out there, as well as to exploit the vast knowledge of the drug discovery process in the organization. So with that introduction, Dr. Ola Engkvist, thank you so much for taking the time to come talk to us today.
Okay, pleasure to be here. So thanks for having me. I should just say that actually since last year, I'm heading a new department called Molecular AI. So it is very still close to computational chemistry, but it has a new name.
It sounds equally cool. So I'd love to start off, as we typically do, by handing our guest the floor just for a little bit. If you can take us a little bit on the early journey, everything from where you grew up, when you first developed your interest in science and chemistry and computational chemistry, and a little bit of your early career journey. I think that would be a great way to start things off.
Absolutely happy to do that. So I grew up on a dairy farm in the very south of Sweden called Skåne. So I lived there the first part of my life and went to school. And I think it was basically in high school I started to get really interested in science. I think before probably history was the most interesting subject for me. And I think based a lot on curiosity, how things work, and particularly chemistry, because I think it's a bit more kind of logical than biology to me. There are still certain rules that you can follow. So studied science at high school, and then decided to continue at the University of Lund, so to say the university in southern part of Sweden. I studied chemical engineering there. In the end, actually took mainly chemistry and math classes. And then there was an opportunity to actually pursue the combination mathematics, computers, and chemistry. So I did a PhD in theoretical chemistry as a subject. So worked on what is called intermolecular forces to basically describe the interaction between molecules as accurate you can do. And then you can use it then to simulate, say, water solution or a ligand binding to a protein. And I really enjoyed it. And that was a very inspiring environment, I think, where I did my PhD, and I learned really a lot. And then I will continue as a postdoc. So I was postdoc for 3 years. I got a Marie Curie fellowship to be in Cambridge for 2 years. Also, I think a very nice research environment and enjoy it really there as well. And the particular atmosphere you have in Cambridge, which I think is something special. And then I spent 1 year at the Josef Hrošč Institute in Prague in Czech Republic, also working on intermolecular interaction more from a biological viewpoint. But then as a postdoc, I started to actually wanted to do something different. At that time, I think computational chemistry was a pretty major field. The progress was in many ways slow. There was nothing much happening, I think. And if an article, say 2000, kind of looked exactly the same how it looked like 1990. So it was, yeah, it didn't happen that much. And I wanted to see and do something different. So I joined a biotech company. And it was when it was like a boom 2000. There was boom in biotech. The first gene was sequenced, and there was of course a boom in a lot of other fields as well through the internet, and that was nice. But as we all recall, the boom around 2000 ended up in a bust. So I worked for 2 biotech companies, but unfortunately none of them survived. And then again, I wanted to do something different. And then I moved to the pharmaceutical industry. So I joined Astra as a staff scientist 2004, joining a technology function, which I'm basically still part of, developing new methods for computational application for drug design. And then been through a few changes at Astra, also some promotions to first lead a group, and now I'm leading a small department.
Excellent. Excellent. And thinking about some of the terms you're using, I think back in my own time in pharma, and think back even longer than that, say the last century or so. Drug discovery has changed quite a bit from very crude accident methods into the era of sort of the natural products, the sort of initialized chemical libraries, into the era of combinatorial chemistry and SAR, and now into sort of the cutting edge of what you're doing. Today in 2021, and obviously not specific to AstraZeneca, but what does drug discovery, if I was to walk in and walk around your area of AstraZeneca, what does it currently look like? Is it many Dr. Ola Engkvist sort of at computers thinking about, or do you still have people actually screening substances and seeing sort of what works?
Oh yeah, no, definitely. I mean, drug discovery and that drug design is fundamentally an experimental science, but because in the end, when you submit a compound for regulatory approval, either as investigational new drug or for a market application, it's fundamentally based on experimental data. So it's very important to make that clear. It has always been and will always be an experimental endeavor to develop new drugs. But we think, and of course the whole industry thinks, that you can definitely speed it up with applying machine learning and AI, and that's happening a lot. There's also a lot of progress in the experimental area or in automation. So you just look at the sequencing. I mean, 2000, it was a tremendous effort to sequence 1 human, and now you can sequence many 100,000, even millions of individuals. So it's been also tremendous progress in automation that then feeds into more use of data science and AI.
And along those lines, you in 2018 you wrote a paper that sort of sort of arching history of AI and machine learning in drug discovery. The title was "The Rise of Deep Learning in Drug Discovery" in Drug Discovery Today magazine. And you talk about sort of there was this first wave of these technologies for looking at sort of bioactivity predictions, but since then it's moved on to de novo molecular design, prediction of synthesis during the process, biological image analysis. Take us, I know this a couple of years ago, but walk us through, if you would, a little bit of some of your core learnings in this paper.
Yeah, so basically, I mean, it's kind of 2018, it's just 3 years ago, but it's been a fast developing field. So actually quite a few things have happened. And so just take the history of cheminformatics and computational chemistry in industry. I think there was also a quick development very early on in the late 90s when there was combinatorial chemistry.
Yep.
And high throughput screening arrived. And then I think that the field cheminformatics started. I think the term was coined 1998.
Okay.
There was a lot of progress there with fundamental algorithms. And then I think not that much happened, maybe for 15 years on the surface. We were using more or less the same algorithm. We had a little bit more computer power and so on, but nothing really significant. But there was a lot of things happening actually under the surface.
Okay.
With the arrival of like open source tools like RDKit for cheminformatics, manipulation and so on, databases like Campbell. And another database is like the GDB database, where they have enumerated all fragments for graph theory.
Mm-hmm.
And then for us, we had an academic collaboration, or actually it was a colleague of mine that had a more or less a chance encounter at a conference of a then unknown Ph.D. student, Marvin Segler. We invited him and started to collaborate with him a bit, and that resulted in a joint paper. It was the first paper describing recurrent neural network for molecular design, which I think has been both influential actually in the industry, but very influential how we are thinking about it. And actually that was only a side project for Marvin. His main project was his synthesis prediction paper, which has been very influential in that field, and he published in Nature. So basically it started for us in 2016, beginning of 2017, that actually there was a method that we could use to search the whole chemical space, which could be like 10 power 60 molecules. Or yeah, it's such a big number, so you can't enumerate it. You can't imagine it. And then we continued to work on that. We did some validation studies that we published together with Professor Jean-Louis Raymond in Bern. We actually showed that we actually could sample the chemical space, and we showed that for fragments where you can do also the enumeration, so you have the ground truth to compare with. And then we have continues to evolve the platform and develop new science. So we have been in several international collaboration, EU funded, to train PhD students. I think that has been very beneficial, I think both for us and for also the academic partners. And then we have successfully improved the tool, particularly around scoring the molecules. Okay. Because I think with the RNN, we have a good way of sampling the chemical space. But then you need to know if your ID is good or not so good. And that's a lot that is depending on how you score them. And we have worked both with machine learning models there to predict properties like solubility and permeability and activity, but also combine machine learning AI with called physics-based modeling. Okay. So what I mean with that is like simply stocking, when you look in a ligand to see if it fits in a protein cavity. Mm-hmm. As we use that for scoring our molecules, and then you have more computational heavy methods that you can then score molecules even better in the post-processing step, like called free energy perturbation calculation, where you calculate so to say through a simulation the binding affinity. So we are continuously developing the platform, and then we hardening it as well. So it's not only to run academic codes. That's always the step to improve it, to harden, so we can use it, and then we are applying it to the internal pipeline. So currently we are approximately supporting 2/3 of the small molecule drug discovery portfolio with a suite of machine learning and AI tools for drug design.
Excellent, excellent. And in a quite recent paper of yours, it was in May 2021 in Theoretical Computational Chemistry. It was a comparative study of deep generative models on chemical space coverage, where it's beyond the scope of this program in terms of sort of the uniqueness of these. But I actually did a show a little while ago where it was with actually an investor that was getting a lot of business plans on the AI and drug discovery front. And, it was one of those things that shocked me how you keep up with, because like anything else, everyone's creating lots of new AI and new models. Based on looking at this type of study that you're reviewing these, you're in AstraZeneca, and you not only have to discover the new molecules, but you got to keep evaluating these new technologies and these new models that are coming out. Talk a little bit about that, if you would. I mean, how much do you spend your time on each of these activities?
Yeah, I mean, it clearly, so to say, in machine learning AI, it comes with a history of open source to publish your results and data, and of course the main, so to say, deep learning engines, they are PyTorch, original developed at Facebook, and TensorFlow, which is originally from Google. And I think everybody's using one of those two. So the barrier to do science, which I think is very good, is low. So there could be a lot of contribution from all over the world. It's not the most biggest and famous academic labs that can do important contribution, but it does create a lot of literature.
Yeah.
And a problem I think that we have a little bit inherited from computer science is there have been a certain focus on beating benchmarks, and I'm not really sure for that as a scientific field if that has been is really that useful. So when I read through articles that should inspire our work internally, I see look more is there any original idea? What is the reason to believe that it works? How will it complement what we are doing at the moment? And so it's my job is actually, there's quite a lot of reading and assessing what people are in the field, and see how we compare, and which collaboration with academics and other organizations are important for us to develop the best possible computational tools we can to support the internal pipeline. But it's a lot of work, and that's good in many different ways. It's not only around accuracy. Speed can also be important, and how much you need to interact with the system yourself. So there's a lot of different parameters you need to so to say include in your assessment. But I think that the field has moved forward quite quickly, and there's a lot of different algorithms that have been tried out. And for instance, what we have published recently is around the transformer architecture, and that I think works very well in our field, like in many other fields. After all, you can describe molecules with a language called SMILES, which have its own grammar. So you can see many times like a chemical reaction or molecular optimization as a language translation problem.
Got it.
So I think that's one reason of the success in the field have been this, so to say, that we can utilize is to say algorithms that have been developed in other fields for our field. So that's, I think, one reason it has moved forward very quickly.
Got it. Got it. And Ola, in addition to obviously your work in molecular AI within AstraZeneca, I've been reading about some of the external collaborations, and one of the extremely fascinating ones is this paper on malaria inhibitors, where you're partnering with Novartis and GlaxoSmithKline, Cambridge, Medicines for Malaria Venture Discovery Group. obviously non-confidential or confidential, but can you take us on a little walk through this project, because it seems extremely timely and very collaborative?
Okay, so that is actually a very old project that was started many years ago through our colleagues, and that was basically to support to be able to build a general model for malaria, and then many companies contributed their data, and then it was actually redone a second time for reason there was some that had actually new data, and so therefore a new publication was published. But actually the original article there, I think, was from 2015 or 16, so that's actually pretty old. But though what we are doing now, that I think that is interesting, we are part of a EU project called Melody. Then we are 10 pharma companies that are doing privacy preserving machine learning. So we don't share any underlying data, but basically by training it with a deep neural network and basically train the middle layers together, we can improve our models without sharing any underlying data. So that is, I think it's a more modern way. I think the Malaria paper is nice, but it's actually based on actually quite something we did quite a few years back.
Got it. And when I saw sort of the pharma names and then the different, it was just a very interesting collaborative model. And any other, once again, non-confidential examples, some cases that anything you want to mention on that front?
So I think it's, I can mention a little bit about our collaboration with NVIDIA. So actually there is a nice video, I think on LinkedIn from NVIDIA with one of my colleagues, Lindsay Edwards, telling a little bit about the collaboration. NVIDIA have a new supercomputer in Cambridge in the UK called C1. And we are collaborating with NVIDIA on developing a giant GPT-3 like chemical transformer. So basically, like, to really see how well we can train a transformer architecture on all publicly available chemical compounds of interest, and then see how the improvement of the model, so to say, changes with size. So that's I think a very exciting project that we are collaborating with NVIDIA on. So and then we have several academic collaborations as well.
Excellent. Well, obviously, we hear a lot about these tools like AI, machine learning in the early stages of drug discovery, but clearly there's a lot of vision in terms of what happens next, because as you know, there's a lot that happens between drug discovery and then me taking that pill at the end of the day after it's registered. Some of your visions, some of your ideas, and obviously we hear about the potential to shorten preclinical drug development activities, maybe a little more futuristic, this whole principle of in silico clinical trials. Going out 5 years, 10 years, take us on a little journey of where you think some of these technologies may be taking us in drug discovery and development.
Okay, so first a caveat that we do think it's still an experimental science, and all clinical decision will be based on experimental data. But I think it's there are a lot of potentials. But what I think is have a very high potential is the combination of new technologies progressing in for instance, transcriptomics, but also cross-genomics, that should enable, together with technologies like knowledge graph, to improve target selection. So we can work on the right targets. I think historically, if you see on failures, of course there could be safety failures, but there have been a lot of so to say, failure in the clinic, in the industry due to efficacy. And I think there, by further understanding the biology through a combination of high throughput biology and machine learning, AI, and data science, I think we can really hone in on, so to say, in the future, much more, so to say, targets that actually translates. So I think that there is an enormous potential for impact. And I think in clinical trials, I think they of course gets more and more complex, and you collect more and more data. So there is a natural component of data science in analyzing the clinical trial and select the patient cohorts and so on. So it will be much more digital. But I think it's still that you will need to generate experimental data. It's not. I think the in silico patient is quite far away, and I think it's a little bit dangerous when people are telling that they will cut the whole drug discovery and development to 1 year. I don't simply think that's not realistic. Simple. You need to generate solid data, and AI and machine learning can help you to do that quicker, to hone in on the most important data that you should generate quicker and analyze it really efficient as well.
Got it. And you just sparked one more thought in my mind, because obviously, 15 years ago, if I was to look at the top 10 selling drugs in the world, they were all small molecules. Nowadays, I look at that list and I don't recognize any of them because they're biologics and so forth. Just a couple of minutes, your thoughts on, whether we're talking about proteins or mRNA therapies or gene therapies or whatever's coming down the pike. What do you brainstorm about in terms of how you can apply your knowledge of molecular artificial intelligence to all this new stuff that is becoming sort of the top?
Of course, first you should say that it's really fantastic. It's really a lot of innovation at the moment. And of course, already in the 90s, antibodies came along, has established itself. And of course, as you point out, most of the top selling drugs nowadays are antibodies. And there's of course a lot of innovation in modalities, and already starting with small molecules. Then you have, say, protein degrader called PROTACs, which I think is a very promising field. One also looks more rationally into covalent inhibitors. It is also, I think, a promising field. But then you have all the so to say cell therapies, that's of course CAR therapy, and so to say nucleotide therapies, antisense oligonucleotide, and mRNA. So there's a lot of things happening. I would say when it comes to data science and AI, it's more challenging there than for small molecules. So I don't think it's as advanced in for other modalities, but clearly there's also an opportunity for the future to do more. And when you generate more data for those modalities to build models, to see what you can do with molecular dynamic simulations to actually understand, so to say, the processes for those modalities. And there's a lot of work going on with, say, for antibodies. How do you represent that best in deep learning? So it's more of an emerging field compared to in small molecules.
Got it. Ola, coming back to you now, obviously you've had a fascinating career so far, big pharma, startups, academia, collaborations across that. Giving the mic back to you now, just for the wrap up, if you want to mention any influencers, mentors throughout your career, your team, whomever, please take the floor and shout out to mention anything that I've missed or anything else that you might want to say about it.
So I think there's a lot of people influencing me, and I think one was definitely my thesis advisor for my PhD. I think he had a fantastic intuitive understanding how nature worked, which I still admire, actually, that and he was very good at teaching it and spent a lot of time with me teaching it. So I think that's something I brought with me. I think it helped me a lot of understanding around molecules, molecular interaction, and so on. And then I think also at Astra there are several inspiring leaders, particularly the current leadership with Pascal and Mena. I think they have a fantastic understanding of the whole drug discovery development process from bench to bedside.
Excellent. It's going to be a fascinating next few years to continue to watch what you're doing in this space, because it is really the cutting edge, and it's just fascinating to hear you talk about it. A lot of it over my head, but nonetheless, it's a very different pharmaceutical industry nowadays and what's going to be coming. So for everybody that's going to be listening to this particular episode on our podcast or watching on the YouTube channel, you've been listening to Dr. Ola Engkvist, Associate Director, Molecular AI, Discovery Sciences R&D at AstraZeneca Pharma. He is also Adjunct Professor in Artificial Intelligence and Machine Learning-Based Drug Design at Chalmers University of Technology. Ola, I want to thank you for taking the time out of your schedule to come talk to us for a little while about what you're doing. Thanks for everything you're doing for the future of health. And as we say on our show, thanks for helping to create a better tomorrow through all your work. Really very inspiring.
Okay, thank you. And thanks for having me.